So we're going to be chatting about the B cell response, first of all. Response that's mediated by B lymphocytes. And this response is called humoral immunity. Greek word humor means fluid, liquid. Is that clear? So humoral immunity is the one that is soluble. Why we call humoral immunity humoral? Let's figure it out first. Humoral adaptive immunity is mediated by antibodies. Does that make sense? Antibodies are proteins, gamma globulins. Gamma globulins can be found in the blood. Does that make sense? So antibodies are soluble molecules found in the blood, in the plasma. This is why we call this branch humoral. Antibodies mediate, soluble proteins mediate this branch. We clear? Now let's chat about how it works. So B cells can recognize pathogen directly. How do they do that? So on the right hand side here, you can see a graphical representation of a molecule that we call antibody. You see that it consists of the two heavy chains, two light chains, and it has this fork-shaped structure, right? You see that fork-shaped thing, you know? So it turns out that each part of the fork has its own important function. The handle, or stem region, is called FC fragment. C stands for constant, sometimes referred to as cellular. The function of that fragment is to bind to cells, to other cells, like cells, macrophages, cells of immune system, basically. Okay? This fragment is how your immune cells see antibody. So, as you can guess, the name constant implies that this part of antibody doesn't change much. We clear? Now this part, sometimes called FAB. AB stands for antigen binding. So there are millions and millions of antigens out there. So FAB fragments of antibodies have to be very variable. Is that clear? And they are, indeed. So basically antibody, it's like, you know, if we think about a fork, think about antibodies as forks, which all have the same handle, but the prongs are all different, all of them. Does that make sense? We good? Now, to kind of clarify things a bit, FAB fragments on the same antibody have the same specificity, meaning that this antibody, if it recognizes, I don't know, flu virus, that this recognizes flu virus and this recognizes flu virus. Does that make sense? Now, if we move back for a second, you will see that is antibody. This is FC fragment. This is FAB fragment. So you see that FAB fragment of an antibody recognizes the antigenic determinant on the antigen. Again, FAB fragment of the antibody recognizes antigenic determinant of an antigen. Is that clear? Now, antibodies, very different. And it turns out that all your B cells, after they mature and, that kind of stuff, they all have different sets of cellular receptors called IgD, which is equivalent to B cell receptor. So this IgD thing, Ig stands for immunoglobulin, D is just, immunoglobulin D. So IgD is a receptor on the surface of the B cell that can recognize the antigen. Does that make sense? So look at this picture and tell me how many different types of B cells can you see if, color allows you to do that. 3, yeah. So we're going to call them like green, blue, and blue? What's the name for that? Huh? Light blue. Okay. Green, blue, I don't know why they couldn't select something like more maroon, for instance. Light blue, okay? So you see that green B cells have certain type of the receptor, blue have different, and light blue have different, right? And each type of receptor can recognize its own unique antigen. So far, does that make sense? You with me? Now, the population of B cells with the same receptor is called a B cell clone. So that's clone 1, this is clone 2, and this is clone 3. So you have 3 clones of B cells in that image. Does that make sense? All cells in one clone are the same. They all can recognize the same antigen. So how many clones you have? Millions, probably. There are very few B cells in each clone, but there are millions of different clones. Does that make sense to you? Now here you see that antigen, shown here in red star, is recognized by only one clone, clone number 2, the blue one. You see, green one and light blue clones, they don't recognize that antigen. See what I'm talking about on the picture? And that recognition leads to 2 events. So recognition itself, that act of recognition, is referred to often as clonal selection. So clonal selection means that that particular clone is selected. Does that make sense? And then that particular clone starts to proliferate and differentiate. Proliferation means that the cells become more and more numerous. Differentiation means that they become specialized. You see what I'm saying? So they differentiate into effector cells, the plasma cells and the memory cells. And plasma cells start to produce antibodies. And you see, look at the color of antibodies. They're blue. They have the same structure and same specificity as the B cell receptor. Does that make sense? Think of it as think of a B cell as a number of what you call them, the bits for the screwdriver. You need to unscrew something. You look at the head of the screw and you match the best bit to it, and you select just one. That's clonal selection. You select a single clone of B cells, right? And then that single clone of B cells after the clonal selection, okay, it expands. That's clonal expansion. But by expansion, we mean you have a lot of B cells of that particular type, okay, that differentiate into plasma cells, antibody-producing ones, and memory B cells. That primary response, does that make sense to you? Now, there's one problem. Your tissues, your proteins in your tissues, can they potentially be antigens? Yes or no? Yes, they're proteins. They can be antigens. They totally qualified for it. So, what happens during B cell maturation? What happens during B cell maturation is that B cells that have a potential to recognize your own proteins are eliminated. Let it sink in. Think about it. So imagine that you have 1 million different types of B cells with 1 million different surface receptors. Does that make sense? And then you test, well, taking it in quotation marks, we're talking abstract. You test each type of B cell, whether it reacts with your own tissues or if it doesn't. If it reacts with your own tissues, that's not good. This clone of B cells will be eliminated. Does that make sense? If it doesn't react, good. Yep, we got the clone. Understood? Now, sometimes that selection, it's called negative B cell selection, because if they respond, if they autoreactive, self-reactive, we gotta kill them, we gotta negate them. So that negative selection fails. If negative selection fails, and your bone marrow starts to release B cells that basically react to your own tissues, what kind of condition are you going to develop? Bingo, autoimmune disease. Say, systemic lupus erythematosus, rheumatoid arthritis. Okay, they have that B cell portion. Got it? Now, what happens during the secondary response? Remember, in the first response, you've got a bunch of plasma cells, and you've got some memory cells. Are you with me? So these memory cells, in your second response to the antigen, will produce much more robust response that will be much faster. Here you see memory B cell that is already primed for differentiation and proliferation when is exposed to the antigen, produces a lot more plasma cells and a lot more memory B cells. Good? So this graph shows you a generic antibody profile, the antibody titers, in the plasma of a patient after the first exposure. So you see it takes some time to start accumulating antibodies. They peak, and then they fade away. And then after the second exposure, antibody to that same exact antigen, they reach new heights, and they reach new heights much faster. That's the booster effect. When you get a vaccine, your first response is fairly weak. So, say, like hepatitis B vaccine, 2 weeks later, you go for a second injection. And that second injection will boost your antibody response. Does that make sense? And then about a month later, go for the third injection. And the third injection will be somewhere here. We clear? Now, the first type of antibodies that is produced during the primary, so when you get your vaccine first time, you first produce antibodies called IgM, and only then you produce IgG, and then antibodies called IgA in the mucous membrane that we'll talk about. So there is a very distinct pattern of types of antibodies that are produced upon the exposure to the antigen. We good? And here we come to the antibody types. So, remember I mentioned IgM. That's the first responder. Okay? Why we pay so much attention to the fact that IgMs are produced first? Let's say you have a patient and you suspect some kind of infection. So you suspect yellow fever infection, ongoing yellow fever. You can draw blood and test the patient for the presence of IgM antibodies. If they are present and they are against yellow fever, then yes, indeed, this patient has an ongoing first exposure to yellow fever. Does that make sense? You got it? Like right now. Now, IgG, being the most abundant antibody, comes second. By the time IgM's gone, IgG starts to climb up. Very often, you will not see the significant antibody titers, significant IgG titers, until the infection is resolved. Make sense? IgG is capable of crossing placenta, and this is how vertical immunity is conferred. You may have heard about the new practice that is implemented in the United States. Pregnant women receive DTaP vaccine. Okay? And the main reason behind it is that getting a DTaP, usually it's a booster, it's not like a new vaccine, right? So that booster increases amount of immunoglobulins G in mother's blood. And those immunoglobulins will be transferred to a fetus, so that when fetus is born, the baby has considerable amount of antibodies that will confer some protection to baby for some time until the baby is eligible for DTaP, their own DTaP. Does that make sense? Okay. IgM cannot cross placenta. However, both IgG and IgM can activate complement. So they both work through the classical pathway. IgA, sometimes called secretory. It's a dimer found in the mucous membranes. Okay. Also in all secretions such as tears or saliva or breast milk. IgA is the key component that protects newborns during the breastfeeding. That makes sense? It's actually really, really important. A bit of a digression. He would look at, so why, I'm kind of telling you the story about the importance. One of the main health problems in Africa, the one that is infectious diseases problems, secondary only to HIV, is malaria. It claims a lot of lives. Now, if you would look at the pattern of infections, the population that is most susceptible to dying from malaria are going to be children at the age from 6 months to about 5 years. Now, why? Why not 1 month or 2 months? I mean, I hate to say this, but those are poor countries. They basically cannot afford formula, right? So what babies receive as a food in the first months of their lives? Mother's milk. And their mothers, most likely had multiple malaria infections before. So they have a lot of antibodies in their blood. And they transfer those antibodies to their babies with the breast milk. Eventually, due to, I don't know, time constraints, work, job, whatever, babies are weaned off the mother's milk and they start to eat regular food, and that usually happens around the age of 6 months. Okay, they receive less and less that antibody protection from mom. And that's when they become susceptible to malaria. Why 5 years? Well, they become kind of stronger physically, so it's just harder to kill them, I mean, it's not limited, like not 5 years and you're fine, but the general saying, in Africa, in malaria abundant areas, if the child makes it to 5 years of age without dying, most likely that will be a grown-up. But a lot of them don't. Okay. Make sense? The idea behind the mother's protection. IgE, allergy. Allergic reactions. That's it. That's allergic antibody. And IgD is the most boring antibody. It's the B cell receptor. So sort of in a nutshell, IgM is the first responder, can activate complement. IgG is the second responder, can activate complement, crosses placenta. IgA is secretory antibody found in the breast milk, saliva, and mucous membranes. IgE is the allergy antibody, and IgD is the B cell receptor. Does that make sense? And what can antibodies do? First and foremost, antibodies do not kill. They do not lyse. Make sense? What they do, they tag the pathogens. So here, you can see a virus on the left being tagged by antibodies. This is called neutralization. So you see, virus is completely surrounded by antibodies. And what can happen next is those antibodies will be in turn recognized by macrophage, and macrophage will destroy the virus. Or nothing. Virus is just surrounded, it cannot go anywhere. Does that make sense? It's like cloaked. Antibodies can agglutinate. Here you see antibody-mediated agglutination of red blood cells, but, honestly, more importantly, it can be a pathogen. So these IgMs here will bind multiple cells, bringing them together, immobilizing them, and making them more attractive for phagocytosis. We good? Do you understand what neutralization, like neutralizing is just blocking, okay? Agglutination, bringing together. Precipitation. Now that is fascinating. Soluble antigens. Now, I try to avoid examples directly from infectious diseases, so let's chat about venomous snakes. If you get bitten by a venomous snake, what do you receive? Antivenom, yes. And what is exactly antivenom? It's antibodies. So it's produced in a fairly simple way. The venom is collected from a snake and then injected into a horse without killing a horse. Of course, the dose is regulated, right? So venom, the snake venom, contains some key antigens, and these key antigens will induce the immune response in the horse. And the horse will produce antibodies against the venom. Does that make sense? Those antibodies are then collected, and the convenient thing is you don't have to kill the horse. Like, you can repeatedly give the horse venom and draw blood, and you will have, as long as the horse is fine, it's going to give you the same antibodies. It's great, And you isolate those antibodies and you keep them as antivenom. And when somebody gets bitten, you give them those antibodies. Got it? And they will precipitate the antigen, the venom, making it dysfunctional and targeting it for phagocytosis. That makes sense? Actual kind of semi-practical outcome of it. You may have read about people in the medieval Europe, training them to be tolerant to poisons. Well, it depends on the poison. Like if you decide to build tolerance against arsenic, you're going to die because arsenic accumulates in the body. But if you will rub a bit of a venom into your skin and you will increase the dose, you can actually build a pretty good tolerance. Again, I have one student that is fascinated by snakes, and he has actual cobra, and he mentioned about some Australian dude who handles taipas, the world's most venomous snake. And he's just like, you watch the video and he gets bitten by taipan. And he's totally fine because he was injecting the venom for years and built enormous immunological tolerance. So basically the moment poison gets into the circulation, antibodies block it right away. Does that make sense? So yeah, it can be done. It just takes a lot of time, and if you're not at risk of being bitten by a venomous snake, there's no point in doing that. Okay? Now, we've got neutralization, we've got agglutination, we've got precipitation, and all of it enhances phagocytic responses. Okay? Now, another way the antibodies can work is that they, IgM and IgG, can activate complement. So complement activation can enhance, again, phagocytosis, inflammation, and also lead to the formation of membrane attack complex that will lead to cell lysis. You see, antibodies do not kill anything. They just basically stick, right? They tag, they attach to the surface. Which another system of proteins that we discussed today when we talked about innate immunity attaches to the complement, attaches to the pathogen surface, making the pathogen more attractive to phagocytosis. Which protein system in the plasma results in phagocytosis enhancement? A bit before. Reference trigger antiviral state. Those, which proteins opsonize? Which proteins opsonize? Which system leads to opsonization of the pathogen? So again, complement. Yeah, complement. So it's quite a while back. But complement here, see, it leads to opsonization of a pathogen. And we mentioned what opsonization is. Essentially, complement proteins, they stick around the pathogen, making it more attractive to macrophages and neutrophils. Does that make sense? Got it? Now, if you look at the mechanisms of antibody response, antibodies stick around the pathogen, making it more attractive for macrophages and neutrophils. Essentially, antibodies opsonize a pathogen. So we have two different branches of immunity. Innate, represented by complement, and adaptive, represented by antibodies. But both these branches lead to opsonization of a pathogen. Both complement proteins and antibodies can opsonize. Does that make sense? Now, humoral immunity can be categorized in 4 groups based on the presence or absence of a pathogen exposure and based on where it comes from. So humoral immunity can be natural and acquired. Natural immunity does not require humans, acquired does. Does that make sense? So if you get an infection, that's natural, right? If you get a vaccine, that's acquired. If you get transfusion of immunoglobulin, anti-venom, is acquired, is artificial, sorry, not acquired, artificial. Does that make sense? Artificial. So natural versus artificial. Does that make sense? Now, there is also, division into passive versus active. Active immunity implies that there is an exposure to an antigen. So again, when you get a vaccine, or you get an infection, or you're bitten by a venomous snake, that's active immunity. When you receive anti-venom, okay, fair enough, you've been bitten. Forget it, anti-venom. When you get antibodies from your mom, either through placenta or breast milk, that's passive immunity. Does that make sense? When you get, after some kind of environmental exposure, you get a tetanus shot. I suppose it's more linked to the injury of some kind, So you get a tetanus shot. Tetanus shot is basically antibodies against tetanus. Okay? That's passive immunity. You see what I'm trying to say? So, say vaccine. Is that natural or artificial? Artificial. Is that active or passive? Active. You get exposed to an antigen. Now, intravenous immunoglobulin. Is that natural or artificial? When you get a drip. Artificial. Human, sticks it into your vein. Active or passive? That's passive because you have no exposure. You just get antibodies from someplace else. Passive immunity does not confer any memory to it. Does that make sense? Now we're going to talk about cellular immunity, and this is where I cut some stuff out. So T cells, right? That's T cell, future T cell, goes into thymus, matures in thymus. And in thymus, all T cells split into 2 large populations, CD4 and CD8. You with me so far? Now, the interesting thing about T cells, B cells can recognize pathogen directly, antigen. There's a statement. B cells can recognize the antigen directly. Does that make sense? So it's like, B cell floats by its antigen, says, that's antigen, I'm going to recognize it. Recognizes it. Cool? T cells cannot do that. Antigen must be presented to them. So if Jamie is B cell, and she would notice an antigen, okay, something foreign, okay, Jamie will say, that's the antigen, I'm going to make antibodies against it. Makes sense, right? Now, if Casey is T cell, this thing can hang around her forever. Nothing's going to happen. T cell needs an antigen presentation. Basically, it means that some other cell has to come over and say, hey, that's the antigen. That's what you are going to be hunting for. You got it? So that antigen presentation is mediated by antigen-presenting cells. The main most important type of antigen-presenting cells are dendritic cells. Okay? And also macrophages and B cells. So these are three types of antigen-presenting cells. Dendritic cells, macrophages, and B cells. You with me? Now, antigen presentation is conferred by proteins called MHC, or now don't feel dumb, I just really want to break it down. Major. What does English word major mean? Big, most important, right? If it's most important, it means that they're less important. That's true. There are less important. Okay, that's most important. H is the crucial here. Okay, histo. Histo. What does histo refer to? Huh? Tissue. Histo refers to tissue. So, the main tissue compatibility. Now, the word compatibility means like compatibility. So, histocompatibility means tissue compatibility. And C stands for complex. So, these proteins serve as the identification of whether the cell is self or non-self. Am I clear? Do you understand what I'm saying? Let's say all cells. I'm one of the cells in the body, okay? One of many. And I carry the marker blue, and every other cell in that body carries the same exact marker, saying, hey, that's us, we belong to this particular body, right? Now, if Natalie would represent a different, organism, then she would carry the green marker. Still normal for her, but different from mine. Does that make sense? So that's my tissue. That's hers. Our tissues are not compatible. That's really important during transplantation, When people, people can be very compatible. They have very similar major histocompatibility complex molecules. So you can transplant between them. Does that make sense? Or people can be non-compatible at all. Very different. Am I clear? Now, imagine that still, I'm the good cell, and voila, I turned into cancerous one. Now I present a different check mark outside, saying, I'm not myself anymore. Does that make sense? I'm not normal. You see what I'm saying? So these MHC proteins, they basically identify the cell as self or non-self.
So when you say like self, you mean like blue, like what you primarily represent. And then when you turn cancerous, that's when you're not yourself.
Yeah, I'm not myself anymore. Does that make sense? So let's say if we would want to kind of make sort of a reenactment, crude reenactment of it, I would give everybody the blue marker first. And you all would be the cells of the same organism, and you're all blue, right? Make sense? And then I would turn one of you and say an infected cell. And I would give one of you, instead of the blue marker, I would give you red marker. So you are different. You not the same cell as everybody else. Does that make sense? Now, there are two types of MHC molecules. MHC1 and MHC2. And it turns out that they have certain differences. First of all, first difference. It turns out that CD4 cells, CD4 T cells, recognize only class 2 MHC. And CD8 cells recognize only class 1. Does that make sense? How can this is called? So this that CD4 cells recognize MHC type 2, CD8 MHC type 1. It is called MHC restriction. And that's hard rule. No exceptions. You clear? MHC restriction. For the fact that CD4 recognize type 2 and CD8 recognize type 1. That's very clear distinction. Does that make sense? So let's say dendritic cell here presents the pathogen to CD4 cell. CD4 cell will differentiate and proliferate, producing memory cells. Okay? And also a bunch of effector cells that we generally call helper. So, generally speaking, CD4 cells are helper cells, meaning that they don't kill or do anything like killing. They help other parts of the immune system to function efficiently. Does that make sense? Now, CD8 cells, when they recognize the antigen on MHC, when dendritic cell shown here presents the antigen to the CD8 cell also forms memory cells and differentiates into effector so-called cytotoxic lymphocytes. Now, the name kind of implies cytotoxic. Kill other cells. Be toxic to other cells. So far, that makes sense. So, sort of in a nutshell, you've got antigen-presenting cells, and they show the antigen to T cells. They show the antigen to CD4 cells through MHC class 2, and they show the antigen to CD8 cells through MHC class 1. So far, you got it? Now, turns out that class 1 MHC molecules can be found only on antigen-presenting cells. And this is what happens. So this is your CD4 cell. It gets activated. What happens to it? Well, first, it forms memory cell. Good? That's understood, right? Then it forms a bunch of helper cells. Some of them, look here, will recognize the antigen presented by MHC class 1 on the B cells and will help them to produce antibodies more efficiently. Others will recognize the antigen on MHC class 2 presented on the macrophages and will activate macrophages. So far, you're following me? And some of the CD4 cells will essentially help to activate CD8 cells, allowing them to develop into functional cytotoxic lymphocytes. usually call CD4 cells the coaches. Think about football coach. No, seriously. For instance, if you watch NFL, you saw a bunch of football coaches, and if you look at their physical shape, they cannot be on the field. They're going to die of a heart attack in like 2 minutes, right? But you take the coach away, and team is gone. Okay? Because coach will consult quarterback what to do, will consult linebackers what to do, will consult receivers and offensive line. Does that make sense? Coach helps them. CD4 cell makes B cells more efficient antibody producers. It facilitates the differentiation into plasma cells. It activates macrophages, targeting them to a particular antigen. And it also facilitates the maturation of cytotoxic lymphocytes. Does that make sense to you? If only we could find the disease in which CD4 cells are selectively destroyed. Are you aware of any such disease? CD4 cells are selectively destroyed. To see what happens. No? It's AIDS. The virus, HIV, infects CD4 cells. And if it is left untreated, CD4 cells are gone. Now, think about this. You get rid of coaches. You have your quarterbacks and linebackers and wide receivers. The only thing you're missing is the coach. Well, if you're missing CD4 cell, then you have virtually no B cell activation. If you're missing CD4 cell, your macrophages aren't activated sufficiently to, phagocytose the pathogen. And your CD8 cells do not develop into the cytotoxic lymphocytes. So it affects humoral response, it affects cellular response, and it affects innate immune response. You see that? Just by getting rid of one type of cells. So CD4 cells are extremely important. Does that make sense? Another important type that is produced that CD4 cells can differentiate into are T regulatory cells. Why I focus on Treg so much? They dampen the immune response. They regulate it. They actually inhibit immune response. Because, well, think about this. When you produce the immune response to some kind of an injury, eventually you need to kind of, dial it down, right, to kind of stop it. That's the function of T regulatory cells. And what do you think happens if T regulatory cell function is impaired, if you don't have enough Tregs or in the proper location? If your immune response just keeps on going, what kind of condition are you getting? Autoimmune disease. It was very clearly demonstrated that in inflammatory bowel disease, T regulatory cells is a hot topic. We know absolutely 100% that in patients with IBD, the number of T regulatory cells in the gut tissue, in the lamina propria of the gut, is much less than in the patients who are healthy. Does that make sense? So they play an important role in regulating and preventing autoimmune response. Okay? So this is CD4. What about CD8s? Well, they are cytotoxic lymphocytes. Now, what do they recognize? Since I'm recording, well, I can actually move over here and use my first row helpers. Okay. So let's say I'm going to play CD8 cell. Okay. So I'm CD8 cell. Casey is normal cell. Okay, regular cell. And Natalie and Jamie are 2 infected cells, red and green. Okay. And when I got the presentation, you see at the bottom here, this slide, I'm going to try to do it upside down. Here, there is an antigen presentation. You see the antigen presentation? The dendritic cell, that's the dendritic cell, okay, and that's CD8 cell. See that? So during that presentation, I learned about this antigen. I was presented this antigen, okay? And the presentation happened through MHC type 1. Are you following this? So I'm ready, I'm all armed, I'm on the prowl. I'm looking for cells that have that same antigen, the red one, on the MHC class 1. And it turns out that every cell in the human body that has a nucleus expresses MHC class 1. Whether it's a liver cell or epithelial cell of the lung, fiber, whatever, doesn't matter. So say liver cell, okay, and this is fibroblasts, okay, good. So I'm walking around, I'm activated CD8 cell, cytotoxic lymphocyte at this point, and I look at MHC class 1 and associate antigen there. What I see, it's normal, so yeah, whatever. I look at MHC class 1 presented antigen here. What I see, not something that I was taught about. Does that make sense? I don't recognize this. I don't know if it's normal or not. I just don't recognize it, so I move on. And here, I see what I recognize now. That's the antigen I learned about. That's MHC class 1 presentation. I'm going to kill this cell. Does that make sense? Thank you. CD8 cytotoxic lymphocytes can kill any nucleated cell in the body because they recognize type 1 MHC, and type 1 MHC is on every nucleated cell in the body. Does that make sense? Cool. How are they going to kill it? Well, first of all, no phagocytosis involved. Okay. Second, if no phagocytosis, what's going on? Turns out, apoptosis. How? Phos-ligand, phos-receptor interaction, or release of perforins that form pores in the target cell, allowing granzymes to enter the cell and cause apoptosis. This phos-ligand, phos-receptor, and perforin granzyme mechanisms, do they look familiar? Which cells use them? Do you remember? Some innate immune cells. Something natural to them. I'm giving you a hint. Something natural in the name. Natural killers, yes. So basically, natural killers are really, non-distinctive killers, while cytotoxic lymphocytes, they're just very specific, okay? But functionally, they're like NK cells. But clear? Now, this is a general overview of immune system. We're going to go over that thing to give you a more broad perspective, and we're going to wrap it up. So, you've got innate defenses. You've got surface barriers. Does that make sense? Surface barrier is clear, right? Chemical or physical. Innate defenses, leukocyte recruitment, all those white blood cells, phagocytosis, complement interference. That's what we discussed. And then you have adaptive defenses that develop later. So you've got humoral responses. And in humoral responses, you're going to get some immunological memory and plasma cells producing antibodies. And here's the interesting thing. Antibodies will attach to a pathogen and they will increase phagocytosis. Am I clear? So this is your link to innate defenses. Does that make sense? In the meantime, cellular response will lead to formation of cytotoxic lymphocytes, okay, and helper cells. And helper cells will help B cells, providing that crucial link between the cellular and humoral immunity. And they will also stimulate macrophage activation, providing the link between the cellular and innate immunity. Does that make sense? So, immune system is really tightly connected, okay? I didn't mention here the complement activation by antibodies. That's another one, right? So, we're going to wrap up immune system, and we're going to talk about some immunopathologies on Thursday. And we're going to cover lymphatic system. Lymphatic system is laughable. On your way out, please take the quiz. And the Scantron, it's on the blood only.